Hello student, myself Shumona from Simmi Foundation. Today we are here to learn about an interesting topic named vaccine and the types of vaccine. The word vaccine originates from the Latin variola vaccine, which Edward Jenner demonstrated in 1798 could prevent smallpox in humans. And today, the term vaccine applies to all biological proportion products produced from living organisms that enhance immunity against diseases and either prevent or, in some cases, treat diseases. And the most common question is what vaccines do? The answer is when inactivated or weakened disease causing microorganisms enter the body, they initiate an immune response. This response mimics the body's natural response to infection. These antigens trigger the production of antibodies by the immune system. Antibodies bind to corresponding antigens and induce their destruction by other immune cells. Type of vaccine. There are 4 types of vaccine: live vaccine, dead vaccine, subunit vaccine, and genetically engineered vaccine. What is live vaccine? These vaccines are composed or of live, attenuated microorganisms that cause a limited infection in their host sufficient to induce an immune response but insufficient to cause diseases. To make an attenuated vaccine, the pathogen grow in foreign hosts such as animals, embryonated eggs, or tissue culture under conditions that make it less virulent. The example of a live vaccine is MMR. Kill vaccine means dead vaccines. When it is unsafe to use live microorganisms to prepare vaccines, they are killed or inactivated. These are preparations of the normal wild type infectious pathogenic microorganisms that have been rendered non-pathogenic, usually by treatment with using heats so that they cannot replicate at all. The oral vaccine and the subunit vaccine. This vaccine consists of an aqueous suspension of poliomyelitis virus usually grown in cultures of monkey kidney tissue. It contains approximately 1 million particles of poliomyelitis strains 1, 2 or 3 or a combination of all the 3 strains. What is recombinant vaccine? These vaccines are produced using recombinant DNA technology or genetic engineering. Recombinant vaccines are those in which genes for desired antigens of a microbe are inserted into a vector. Different strategies are using the engineered vector that is expressing desired antigen as a vaccine. The engineered vector that is yeast is made to express the antigen such as vector is grown and the antigen is purified and injected as a subunit vaccine. Now we have to know about the DNA vaccines. The DNA is injected into the muscles of the animal being vaccinated, usually with a gene gun that uses compressed gas to blow the DNA into the muscle cells. DNA can be introduced into tissue by bombarding the skin with DNA coated gold particles. It is also possible to introduce DNA into nasal tissue in nose drops. DNA vaccines have induced both humoral and cellular immunity. Now we have to know about cocktail vaccines. A cocktail vaccine is a mixture of different vaccines which is effective against more than one disease. For example, MMR vaccine is effective against mumps, measles and rubella virus. MMR vaccine was first developed by Maurice Hilleman. MMR vaccine consists of live attenuated virus of three diseases administered via injection. So student, in this video you can know about what is vaccine, the types of vaccines and how vaccines are controlled disease. So student, for today that's all. Thank you and keep learning.